everyone. Pull up the lecture. All right. So we'll spend a couple of days doing drugs. We're not going to get into all of the drugs. There are way too many of these, but the analgesics and stuff where people try to remove themselves from a situation. So you go to the weight room, and most people don't go to the weight room with no headphones and just feel it, right? That's usually not what people do. People show up at the weight room and they put in headphones and they try to distract themselves from being there. Like, let me see if I can disassociate from this place, from this emotion, from this sensation. And we do that a lot in life, and drugs are a way of helping us to remove the emotion and the pain and the sensations of a situation. People are like, oh, I'm just self-medicating, and people are using whatever it is, alcohol or weed or something as a coping mechanism. It's sort of the opposite of mindfulness meditation, this mindful meditation where you have an object of attention. It's like a honking car, or it's your breathing, or it's like a pain or whatever, and you just, this object of mindfulness, and you pay attention to it. People sort of do the opposite in life, whether it's with music at the weight room or it's with drugs in their mouths. And so we'll get into some of this stuff. We'll do CBD today. We'll at least start in on CBD, and then we'll finish cannabis, with adding in THC on Friday. And then we'll probably do some other stuff, ibuprofen, certainly, maybe Tylenol and ibuprofen on Monday. And then we'll do altitude, physiology, low, high, and outer space. And then it's a bunch of sleep and disruption of circadian rhythms and shift work and stuff like that. So let me pull up the chat box. All right. So chat box is open. Speak up or type up, whatever, if you have questions. This stuff we'll do last day of chatting about alcohol, just to wrap it up, to provide some closure on alcohol. Been around for a long time. We'll finish our alcohol talk about a little bit more history, but fermentation, how we're going to go from pyruvate to ethanol. Now, for us, we will use that alcohol dehydrogenase to convert ethanol back to acetaldehyde. But in yeast, right, in a brewer's yeast, what we're going to do is create the alcohol, go the opposite direction there, using the alcohol dehydrogenase to create the ethanol. Why, right? Questions of purpose. I have no idea. It's fascinating, but I have no idea. But it's a fungus. These things, they reproduce asexually, and it's a little carbon chunk, right? It's a little carbon chunk. Rubbing alcohol, add another carbon. Ethanol has two. Isopropyl alcohol have three. Methanol has one. But these alcohols, small carbon chunks. There's the ethanol, there's the one you want to drink, that hydroxyl group, right? The OH, the acetic acid. Again, that's the thing that's giving vinegar its vinegarified, whatever taste. And that OH right there, that hydroxyl group, makes steroid alcohol. Cholesterol, a steroid alcohol. You know what distillation is. You know how to create, how to separate, right, the ethanol from the fluid. The azeotropic limit, you know what that is. You have this fluid at the you're heating this fluid and you're evaporating it off, and there's a point at which the vapor and the fluid that's being heated have the exact same composition. That would be the azeotropic limit. Again, ethanol is the one you want to drink. If you're drinking methanol and any amount of bulk, you're going to have a problem. Gastric emptying, right? Ethanol and gastric emptying. It's not going to empty as fast as water will, but by itself, it empties fast enough. But if you have food with it, you're going to slow down the emptying of alcohol. And if you have alcohol with your food, you will slow the emptying of that food. And the longer it sits in your stomach, it's possible that you can break down some more of it. You can metabolize some more of it using the alcohol dehydrogenase that exists in your stomach to convert it into acetaldehyde. If you just keep it there, because you don't just hang on to it forever. You have alcohol dehydrogenase in your stomach, you have alcohol dehydrogenase in your liver, and you will metabolize it at both of those destinations. And so the more of that enzyme you have, the higher concentration of alcohol dehydrogenase, the better you're going to metabolize in your stomach. And so the less that is going to get into your systemic circulation, the less that's going to cross your blood-brain barrier, the less drunk you will get with the same volume of alcohol. Remember, early in life, men tend to have more. Later in life, it's the same. Basically, it's about the same. This is just men over here. With alcoholism, is going to bang up your liver a little bit, and people will be able to metabolize less alcohol, so the alcoholic cannot drink the non-alcoholic under the table, right? If you have a drinking competition, then go against an alcoholic because you'll win. You could be like unadapted and go against an alcoholic, and you'll win because they have damaged their liver enough to where they can't metabolize it very effectively. You know what the flush syndrome is, that aldehyde or acetaldehyde dehydrogenase is kind of crappy. In the place of a glutamate, there's a lysine, there's a polymorphism. The gene itself is a little bit different. And so it codes for a protein that has an amino acid swapped out of it. Remember, we have these 20 amino acids with which we make our proteins. And if you swap out one amino acid for another, it's not the same thing, right? The shape of it is a tiny bit different. The shape of that protein, it folds a tiny bit differently. The shape is a tiny bit different, and those shapes determine their functions. And so if you swap out a glutamate with a lysine, you're going to get this kind of crappy version, this dysfunctional, crappy version of aldehyde or acetaldehyde dehydrogenase. And so you're not going to dispose of it as effectively. Now the mTOR, we're going to talk more about mTOR as we get into CBD, as we get into THC, as we get into ibuprofen. We'll talk about mTOR and these drugs, of how they affect our metabolism, that we ingest or inject or otherwise expose our tissues and systemic circulation to these drugs. And what is the stress response? What is the metabolic response? How do we react to these drugs? And so making sure you have the foundation of mTOR down. You don't need to know the whole thing, and we aren't really going to spend too much time talking about this side, which is the mobilization of the mTOR enzyme, the mTOR complex, to the lysosome. You take that mTOR complex and you deliver it to the work site. So in the age of COVID, we're all like working online and stuff, but when the world isn't shut down, when the world isn't in quarantine, there's a work destination. I got to go to campus to teach, and mTOR has to go to the lysosome to translate. There's a work destination. It has to get there. Amino acids are how it gets there. You have to introduce amino acids to get it there. But a lot of the regulation, this is sort of a nutritional regulation, but a lot of the regulation, the bulk of it, is on this side, which is, if insulin binds to its cell surface receptor, and then you're going to activate PI3K after insulin receptor substrate and stuff. There's a bunch of steps, but PI3K, phosphatidylinositol 3 kinase, and then you're going to activate PKB, protein kinase B. Protein kinase B is going to deactivate tuberin. Tuberin would be suppressing Reb, and so if you deactivate the suppressor, then Reb can activate mTOR, and then you're doing all your hypertrophy. So we're going to keep talking about how these different drugs interact with that stuff. There's tuberin, TSC2, tuberous sclerosis complex 2, tuberin, which is what it's really doing is Reb's GTP, guanine triphosphate. It's hydrolyzing it to be GDP, but Reb GTP is what binds to mTOR, activating it. Then downstream from mTOR, remember we have P70S6K, or S6K or S6K1. That's a kinase. A kinase phosphorylates stuff. This is ribosomal protein S6 down here. So this is let's turn on translation over here. Over here is let's turn off the thing that's turning off translation. So just like tuberin is turning off the thing that turns on this, this is turning off the thing that turns off that. There's double negatives. When you get into amino acids, there's like quadruple negatives. Get into the gator complexes, stuff like that. When you eat specific amino acids, the recognition of those amino acids, you start getting into like quadruple negatives. It gets hard to keep track mathematically of the mobilization of mTOR, but upstream from mTOR, we're turning it on with PKB, shutting it off with AMPK. And tuberin is one site, and then Raptor, AMPK. Another site would be Raptor, which is recruiting those downstream targets. Regulatory associated protein of TOR, RAPTOR. TOR, target of rapamycin. Regulatory associated protein of target of rapamycin. Not myosin, but mycin. So protein synthesis, remember you introduce alcohol, this is ethanol over here, and protein synthesis is lower, and this lasts, here's 12 hours later, we're still seeing it lower, and there's no ethanol left. There's probably no acetaldehyde in there, but the damage is sort of done for a while, and it lingers. And you can see these phosphorylation states on things like PKB, AKT, or PKB, exact same thing. Remember, that's the thing that's going to phosphorylate. PKB is the kinase that's going to phosphorylate tuberin, shutting off tuberin so that Reb can activate mTOR. Now mTOR is phosphorylated, is activated, and mTOR is a kinase, and it's going to phosphorylate P70S6K. Now that thing is phosphorylated. And so looking at these phosphorylation states, after stimulation, after get some mechanical loading to a tissue, stimulate a tissue. And what are the phosphorylation states? Well, much lower if you are in the presence of ethanol compared to the control group. Non-ethanol here, ethanol here. So the phosphorylation states of these anabolic things, these very protein synthesizing things, the phosphorylation states are compromised in the presence of ethanol, and this lingers. This can last 12 hours. Now, it depends on your behavior, it depends on your diet, it depends on a lot of things. Again, this is one study. It's a convincing, compelling, important study, but it's one, right? And this just has one context. If you are an athlete and you're also an alcoholic, that's a different context. If you're an athlete and you sporadically drink, that's a different context. If you're an athlete and you're 20 years old and then you have ethanol at 9 a.m. and you exercise at noon, that's a different context. So any of these variables are going to change the translational capacity and efficiency that you see. But in general, we assume that ethanol is going to be hard on translation. The ethanol, in the presence of ethanol, synthesizing proteins, tissue regeneration, recovery, these things are going to be hard, even in the presence of mechanical signaling, which goes through different signaling cascades, goes through different mechanisms to activate mTOR than does traditional endocrine and immune system kind of chemical and hormonal activations of mTOR are a little bit different from mechanical activations of mTOR, and ethanol seems to suppress all of it. Ethanol does seem to have an effect on all of it. And the mechanisms, it's probably, and this might not be the whole picture, but it's probably through Raptor, right? Raptor is recruiting the downstream targets of mTOR complex 1, and maybe also there's some 4-EBP1 inhibition. You do tend to see a little bit of extra mTOR complex 2 activation downstream from that. You're getting PKB activation. So you do see in the presence of ethanol a little bit more PKB activation. So there's other anabolic stuff that PKB is doing. That's not just this. That's not just tuberin. Um, but it's shutting off this. So you can get PKB activated and then sort of shut off this. There's fascinating stuff that we'll talk about in 248 with like Depter, where it has this kind of oh two-sided personality of a little bit catabolic over here, a little bit anabolic over there. So if you're activating PKB, it's not the only thing PKB is doing is not it's not just mTOR, but you are shutting down mTOR at the same time. And this is just saying that 4-EBP1, maybe there's a little something more than just the raptor going on that preventing that association with raptor. Maybe there's some long-term stuff going on. Mice, a lot of people use mice, especially when you start getting into things like cannabis. For the longest time, up until like right now, up until very recently, the NIH had these funding prohibitions. If you do cannabis research, or you looking into THC, well, then you are not eligible for funding from the NIH, the National Institutes of Health, the largest funding organization in the world. And so a lot of we'll get into it today. We'll start getting into it today, at least. The CBD and THC research is weird and conflicted and kind of bad. We know a lot about alcohol, and we're learning more and more about alcohol's metabolic implications, its mTOR interactions, anabolism versus catabolism. We're learning more and more, but we're a little bit oblivious, a tiny bit blind and a little bit oblivious to the effects of CBD. I'll talk about them, and the effects of THC. I'll talk about them, what we know, but it's sort of weird and conflicted. And then remember that in grapes, and so obviously in wine, there's resveratrol, and resveratrol is going to help LKB1 activate AMPK. There are drugs that do this. So I mentioned ICAR, AICAR. That's an AMP mimetic. It's a mimic of AMP. Um, it binds to AMPK and helps it get activated. So LKB1 can go do its activation. But there's lots of things that are going to help turn on AMPK. If you're an aerobic athlete, that's what you want, because you get mitochondrial biogenesis, you get better energy, better catabolic consumption of energy substrate. I mean, there's a lot of good things are coming from AMPK. Anabolism is not one of them, though. Anabolism is the opposite of what it does. It's very catabolic. Remember, there's two sites of inhibition for mTOR. Tuberin is promoting tuberin so that tuberin will shut off Reb, so Reb can't activate mTOR. And it's inhibiting raptor so that raptor isn't recruiting a downstream target, so mTOR can't phosphorylate them, so you're not going to get to translation. So two sites of interruption on the mTOR side and a couple of sites to turn on protein degradation at the same time. So if you have something that is turning on AMPK, aka resveratrol, which is helping to do it, it's not so potent, but it is helping to do that, then that is an inhibitor of translation at the same time. Remember the taste stuff. That we have there could be other compounds within the alcohol that's not just ethanol and water, ethyl acetate, something like that, where you have ethanol, the hydroxyl, right, that OH group on ethanol is replaced by an acyl group from carboxylic acid, something like that. You have these other compounds that can find themselves in the alcohol, and that will affect your flavor. We think of vodka as nothing but water and ethanol, and yet the mouthfeel is different, the taste is different, the I don't know if there's an aroma that's going to be different, but there's a difference in the quality and the swallowability, let's say, of uh, vodka, of any of these alcohols. And you get the alcohol percentage up high enough, 44% or higher. That's the range that gets reported. And those clathrates, those cages that sequester the alcohol inside, they trap the alcohol inside. Those waters form this little cage, this little prison. So there's structurability that changes in the presence of more of a higher alcohol percentage, alcohol by volume. As that percentage increases, the structure of that fluid, the chemical structure of the fluid can change. And maybe that affects flavor, but more likely there's a mouthfeel, whether it's watery or it's a little bit fuller bodied, stuff like that can change. The hangover, Visalgia, Elpenor's syndrome. Remember, Elpenor was from the Odyssey. And but Visalgia is the medical name for a hangover. Like if you go to the hospital and you're just super sick and what they write down on your chart is Visalgia, that means they think you just had too much to drink last night. And they think you are like phylloglobalized, right? You're just pretending like you didn't have too much to drink. You're coming in here pretending like you're sick when that's what it is. So that's if you encounter these words, that's what they mean. Now you remember vasopressin. We talked about vasopressin in the hydration section. It used to be called antidiuretic hormone. Vasopressin was ADH, anti-diuretic hormone, but alcohol dehydrogenase is ADH. It starts to get confusing if everything is ADH. And so vasopressin or arginine vasopressin sometimes it's called, but vasopressin, this oppresses diuresis. This reduces, obstructs diuresis. Vasopressin does. So if you block it, alcohol is blocking vasopressin, then you're just letting all your pee out. I mean, so vasopressin is exerting its action. You see over here. At the distal tubule and the collecting duct, it's exerting its action, and but that's it's also doing a little bit of vasoconstriction, but that's keeping the fluids in. If you block it, you're letting those fluids out. And the half-life, it's not that long. Now, people will continue to drink for, it's not like you take 3 shots and that's it and you're done and you've metabolized all your alcohol and it's out of you and the alcohol's in you for a while. You probably didn't do all your drinking in a 3-second period, and you probably didn't metabolize all that alcohol in a 5-minute period. So this inhibition of vasopressin is likely to be sustained, and the actual half-life, I mean, I don't know, 20 minutes, 10 minutes at the fastest, maybe 35 at the longest, but somewhere in that 20-minute phase, something like that. It's going to be the half-life of your vasopressin. Now, you will get dehydrated. Drinking higher percentages of alcohol, you will get dehydrated. You can offset that by just hydrating. But acetaldehyde, the toxicity symptoms of it, remember, it's similar to being hungover, but you're not going to have much acetaldehyde in your system when you are hungover. Now, the methanol, right? Get 1 down, 1 carbon down from your ethanol. Rubbing alcohol, isopropyl alcohol, rubbing alcohol is 3, ethanol is 2, methanol is 1. 2, you want that Goldilocks zone for your alcohol. So if you have methanol, alcohol dehydrogenase converts into formaldehyde. Formaldehyde gets converted into formic acid by the aldehyde dehydrogenase. And remember, that's ant venom. That's not the stuff you want in you. You don't want the ant venom inside of you, and it's going to inhibit cytochrome C oxidase. That's the last enzyme in the electron transport chain. That's transporting it's attaching the electrons to the oxygen that you breathe, and so that transfer of electrons to the oxygen you're breathing, if you obstruct that, cyanide does the same thing. If you obstruct that, you have some catastrophic metabolic problems to cope with. Now, methanol, it's in stuff, right? I mean, if you eat a kilo of apples, if you just sit down and it's like apple season out and you have like a contest and you eat a kilo of apples, you might get a gram or two of methanol in there. I mean, it's not like this doesn't exist anywhere but bad alcohol. It exists a little bit, small amounts in wine, right? There's some small amounts in wine, but this exists in the world, methanol. You just don't want big doses. A tiny dose is tolerable. You can metabolize a tiny dose and not pay a metabolic price for that. A big dose, that's gonna hurt. There's the first Iron Mike. Mike Tyson is who we think of for Iron Mike. In the 1980s, the sort of like greatest boxer of all time. But the first Iron Mike was like 1920s, and he was this homeless drunkard in New York City, maybe into the 30s, early 20th century. The first Iron Mike, his name was Mike Malloy, Malloi. And he was this Irish guy who really was a good drinker. And I'm sure there's articles all over about him. You can look up a fuller story than I'm going to tell. But there was a life insurance policy that some of his, we'll call them drinking buddies, some of his pub collaborators were trying to cash in on by murdering him. And they just kept trying. And the next day he'd be back in the bar drinking, right? And they'd try to kill him again. And the next day he's back in the bar drinking. And one of them, they eventually did succeed in it was like half a dozen times or something, and finally they succeeded in killing him. But one of the attempts was methanol. They're just doing whatever they can to kill this guy, and really horrible people. But methanol, like, yeah, maybe we can get through this. Maybe we can survive this. And what's a good strategy to survive it? We'll just ram a bunch of ethanol in you. Selective metabolism of that ethanol, and and you might be able to excrete some of that methanol without converting it into formaldehyde and formic acid. So the hangover, mostly inflammatory, it's the primary culprit, is going to be inflammation. Now, maybe there's some other stuff that can affect you. There's stomach irritation, right? So alcohol irritates the stomach lining. You're going to create a little more acid in your stomach, change the pH a little bit, and you can get some nausea. Okay, that might contribute. Disrupted sleep, right? If you drink alcohol, you might feel like you fall asleep better. You sort of like drink yourself to sleep, but the quality of your sleep is horrible. You're going to wake up in the night. You're not going to actually get into your nice REM and deep sleep cycles. So disrupted sleep. Okay, that's going to contribute a little bit to feeling like shit the next day. Vasodilation effects might contribute to a little bit of headache. Okay, there are some other contributors here. Inflammation, though. Inflammation is going to be the main cause, the main culprit of this stuff. And just for this study, just looking at like, well, here's your ethanol at no hours, right? And then at 1 hour, okay, we're getting pretty high. We're metabolizing some. It's 4 hours later, 12 hours later, there's just a little tiny bit of this stuff. Your acetaldehyde, there's a little tiny bit of this stuff at 12 hours. But you see IL-6, interleukin 6 is going to go up a lot, right? So it's like the 1 hour, here's the 12 hours, right? Here's 0 hours, go from 0 to the 12. That goes up a lot. TNF, tumor necrosis factor, that's usually just listed as TNF these days, but TNF alpha, tumor necrosis factor, this is just another inflammatory mediator, and the interferon gamma, stuff like this. But the major things they saw in this article was the interleukin 6, IL-6, and the TNF alpha, especially the IL-6. There again, you see the IL-6, these different time points. So at nothing o'clock or whatever, 0 minutes have elapsed, and then half an hour, 2 hours, 4 hours, 12 hours, that IL-6 just goes up and up and up and up. Isoprostanes, what these things are is they're prostaglandin-like compounds. Isoprostanes are prostaglandin-like compounds. Arachidonic acid It's a 20 carbon fatty acid. It lives in your phospholipid bilayer. It lives in a cell wall. And if you can extract that thing, convert it into um, PLA2, phospholipase A2, that's the enzyme that releases it, and then COX cyclooxygenase converts into prostaglandins. But peroxidation of this, if you have free radicals, those free radicals can steal electrons from this fat. And so peroxidation of this fatty acid, and you'll get isoprostanes. And so that's a combination of free radicals, reactive oxygen species, right? If you're like, what do you take, vitamin C for? Well, that's my antioxidant. Yeah, for to combat free radicals. And so if you have a bunch of free radicals, you're going to get more isoprostanes. And so you do see a free radical response also with more isoprostanes increasing. And so this is the overall look at what's going on. Really, it's a giant inflammatory response, but there's a little bit of free radical stuff going on, some depletion of some antioxidants, C-reactive protein. Remember, that's the thing that the liver makes. It's in your systemic circulation, and after IL-6, you're going to get a big release of that stuff. So it's this sort of it's triggered by IL-6, made from macrophages and T cells, and you'll get after IL-6, you get this release of C-reactive protein, this sort of a measurement of overall inflammatory response. So this is really your hangover right here. It's a big inflammatory response. And you start with ethanol, that IL-6 gets going, get that IL-6 going, but also TNF alpha. TNF alpha is also contributing a little bit. There's going to be some other stuff, maybe a little bit of neurotransmitter stuff, maybe some mitochondrial dysfunction. Again, it could be the stomach, could be the sleep, could be blood pressure. So there's other variables that are on the hangover team. Other variables are exacerbating a hangover and contributing, but man, that inflammation, that's really leading the charge. And people have blamed acetaldehyde a lot. Acetaldehyde has gotten a lot of negative attention, in part because of like flush syndrome, and if you just give people a bunch of acetaldehyde, it doesn't feel that good, but it doesn't cross the blood-brain barrier, and so this is very unlikely to be, and it's so low by the time people are having a hangover, unlikely to have anything to do with it. Is it going to be water? No, it's not going to be water. Ethanol is really what's the culprit here. Is going to be ethanol in the hangover. Dolphinimic acid. Remember, this is from the 80s. This is a NSAID, a non-steroidal anti-inflammatory drug. And so we were just talking about isoprostanes as coming from arachidonic acid, that 20 carbon free fatty acid in there's no free fatty, but 20 carbon fatty acid in a phospholipid bilayer. That's where icosanoids come from. And PLA2 is gonna release that thing, phospholipase A2, or is gonna release your arachidonic acid from your phospholipid bilayer, and then you're going to convert that arachidonic acid to prostaglandins with COX cyclooxygenase. It's an ACE, it's an enzyme, converts arachidonic acid to prostaglandins. Now, NSAIDs, non-steroidal anti-inflammatory drugs, they stop that. They stop the conversion of arachidonic acid to prostaglandins. So you stop getting prostaglandins, but you can't wait until you're hungover to take your NSAIDs. That doesn't work. You already have them. It's too late, right? You can't sign up for car insurance after your car was totaled. That doesn't help. Your car is still totaled, and now good, you have car insurance now. You don't put the alarm on your house after the burglar, right? You don't get your stuff back that way. You have to be a little bit prophylactic here. And so that's what this study did, is everybody took their NSAID, their prescription NSAID, their serious NSAID, and they did it before. It's sort of their pre-gaming with it. And then what they see is a reduction in overall symptom severity. And there's a bunch of different symptoms that they were looking at, the headaches and tiredness and vomiting and diarrhea and whatever. And this is just an overall evaluation. Is this, was it effective? Yes, compared to a placebo. And people still talk about this today. It's not like, well, we studied this thing in the 80s and everybody's dropped it. Nobody touches this stuff. It's still being studied today in cancer patients, not because cancer patients are just drinking away their symptoms, but there seems to be making radiation more effective. So there are investigations into concomitant treatment, into adjunct treatment of using Dolphinimic acid for radiation patients, but seems to work really well if you pregame with it as you go into as you're readying yourself to have a hangover. The prickly pear cactus, you remember this one, same thing. They did a really similar study here, and a lot of the symptom inventory. There's a bunch of symptoms, and some of them were better. Overall symptom index, yeah, it's probably a little better. Probably helps a little bit, and it's just going to be a anti-inflammatory. You're going to see this
Call from potential spam.
I'm hearing like a phone ringing.
My bad.
So, but yeah, so baseline versus the prickly pear, right? There's C-reactive protein was lower, and then the cortisol was higher. But C-reactive protein, remember, that is a indicator of systemic inflammation. You get a lot of inflammation going, and C-reactive protein is going to give you a glimpse at that. So wrapping up all this hangover stuff, people have a lot of different explanations of what is causing these hangovers, and everybody has a lot of different cures: the family cure, the fraternity cure, the whatever cure, the personal cure. And some people will say, oh, have a Bloody Mary in the morning. That's one of the things people have, or the Bloody Maria, which is you replace Mary's vodka with some tequila. And that's not going to be helpful. Adding more ethanol when the ethanol was a problem to begin with. Now, it may delay, it may postpone some of these symptoms of the hangover. It's possible to postpone some of these things. And people may be experiencing that, but nowhere in any data have I ever encountered anything that says that's an effective treatment. In the late 80s, propranolol was studied. That's a beta blocker. That's going to keep your heart in check. If you're having, let's say you're a concert musician, you're like a pianist, or you're I don't know, violinist or something, and man, you better not hit a wrong note, or your career's over. You're tense, right? You got to keep calm. If you're having shaky, nervous hands, you're going to hit a wrong note. Propranolol, wonderful for that. Target shooting, propranolol, wonderful for that. If you're nervous and you're like, your hands are shaking, you're going to miss the target by a foot. Propranolol has been shown in the literature to be wonderful for things like this. If you just have a job interview and you're really you don't want like the vibrato voice and sweaty palms and whatever, propranolol, you beta blockers, right? Wonderful for this stuff. But was it helpful for a hangover? No, it didn't do anything, right? You just published, there's a study, 87. There's a study on propranolol and hangovers, and it just didn't do anything. There's vitamin B6. If you take two B6s and sort of shove them together, pyretinol, if you it's two B6s together. There was a study on this that actually showed some effect. 1973, it showed a little bit of an effect, and they had this like a symptom inventory. There were 20 different symptoms of these alcohol symptoms, and they had people do Likert tests on them. On the scale of 1 to 10, what is your severity on X, Y, Z, 20 Zs? And it was 50% improvement. I mean, it was cut by half, but that was in the 70s. Nobody's ever replicated it. Nobody had an explanation of it. And so I don't know. In Courtney's list of hangover cures, is that one of them? But not really. I mean, it may be. It's not going to be harmful. If anything, it'll help, but it's not going to make things worse. And maybe it helps. Maybe it helps. But go ahead and throw that in your cocktail if you want. I wouldn't put in like the tequila or the vodka or whatever in it. Ruin tomato juice in some other way. But sure, throw in some B6 in that if you like. A couple of studies also came out in the 70s on sugar and saying, okay, is it glycemic control? Do we have some hypoglycemia? Nah, neither of those studies found anything about that. So the Jolly Ranchers and you just need Gatorade and you need whatever, none of that stuff. When they have tested that in the literature, it's come back falsified. It's come back negative. It's come back and said that doesn't do anything. So beta blockers, sugar, the whatever, have your margarita in the morning or whatever, that stuff, that's not helpful. The inflammatory interventions, anti-inflammatory interventions, if you pregame with them, that's the thing that actually works. That's the thing that does something. Now, to really wrap up alcohol and its hangovers, we've known about this for a really long time. This isn't new. This isn't something that contemporary society has just begun to cope with, the morning after a hard night's drinking. So this is from Isaiah 5, chapter 5, verse 11. Woe, from the King James version, right? So this is the 1611. It's not the first English version of the Bible, but sort of this really mass-produced kind of poetic version in English of the Bible. Woe unto them that rise up early in the morning, that they may follow strong drink, that continue until night, till wine inflame them. So Isaiah, so this is just English was 1611, but Isaiah, that's Old Testament. And he lived, that was the 8th century BC. Now, was it written in the 8th century BC? Maybe some of it, and it was sort of updated and vernacular and some words and stuff updated for a couple of centuries or something, whatever, who cares? Let's just say 8th century BC that was written, that chapter. That's weird, man. People have been hung over for an awfully long time. And we talked about Odysseus. And Elpenor, right? Iliad and the Odyssey. And the Odyssey, the first English version of the Odyssey came shortly after 1614. It was 3 years later where the Odyssey came out. And also 8th century BC, it was written in Greek. So really the same timing of these books. We have Elpenor falling off the roof with his Elpenor syndrome and breaking his neck and dying and whatever. So this is not new. I mean, this is like 8th century, some of the earliest writings we have, 8th century BC, some of the earliest writing we have is about being hungover and how to cope with that. What's new, is not in Iliad and the Odyssey and whatever, a Testament and stuff, is the other drugs, right? Heroin and cocaine and amphetamine and, ecstasy. And, so this is, we'll talk about a couple of these things, but this study came out in Lancet in 2010. So it's a decade old, but Lancet, one of the top journals in the world, really good article. And it's ranking the harm of drugs, right? And it's multi-criteria decision analysis. So harm to the user, that's in blue. Harm to other people, that's like you're messing up other people. It's like drunk driving. So alcohol is the single worst drug in the world, is worse than heroin. In terms of net harm to the globe, right, to living humanity. If you look at harm to oneself, the crack and heroin, those ones are really bad, and methamphetamine's pretty bad too. Alcohol isn't as bad as those. Now, the stuff that people often say, like, oh, that drug is so harmful and horrible and whatever, like, okay, mushrooms, like that is just, that's like Cheerios. LSD, ecstasy isn't that bad. There's your anabolic steroids. What are people talking about when they're saying all of the, like, oh, these drugs are so harmful and they need to throw people in prison for these things? Like, okay, well, there's tobacco, There's alcohol over here, the worst drug in the world, And so if we're actually objective, if we use our brains and we use science and we're like rational as opposed to offensively emotional, if we actually use our brains and look at science, like, oh, come on, what the hell are we talking about with like steroids and stuff? I mean, look at the stuff that's like actually like doing the harm. Um, and so, ecstasy is pretty low. I've never done it, but I've heard great things from the people who have. The presynaptic release, so from before it gets released, of serotonin, of norepi, of dopamine, that's what it's doing. It doesn't seem to carry too much harm there. Anabolic steroids I talk about way too much in other classes. Cocaine and amphetamine, and these things are really similar, where amphetamine is both the release and inhibition of reuptake. You have to clear this stuff somehow. So inhibition of that reuptake of dopamine in the central nervous system. And okay, maybe that's getting a little bit harmful. Cocaine is inhibiting the reuptake of that stuff, of serotonin, of dopamine, of norepinephrine. It's inhibiting the reuptake. And so a lot of these neurotransmitters, the drugs of abuse, the drugs that people will use for whatever, to offset the lethargic feelings of alcohol, or to stay up for if you're like Jack Kerouac and you want to write for 3 days straight, sit at the typewriter for 3 days straight or something like that. If On the Road, whatever. He didn't actually like write the whole thing. And the legend of writing On the Road, Jack Kerouac, that sort of great American novel of the Beat generation of sort of San Francisco. There's like evidence of previous books, doesn't matter. But so there's a lot of drugs that are going to have these neurotransmitter effects in here, but we're going to focus on cannabis. And there's a couple of different compounds that we'll look at. CBD is the non-psychoactive one. CBD is the one that is not psychoactive. THC is the one that gives you the giggles or whatever. And so we'll focus on this one. Now, I think harm to others, I'm imagining that's probably like stoned driving or something. Because when you look at harm to others with alcohol, I mean, there's only so many bar fights that can happen. Uh, this is probably a bunch of like drunk driving accidents. I mean, nobody, like if you're on mushrooms, everybody just pulls over on the side of the road. And it's like, oh, there's a tree in 6 miles. I don't want to hit it. I better pull over and take a nap. I'm reading that comment box. It would be interesting if they conducted the study in Amsterdam, where all drugs are legal. Curious to see how the results would differ. So yeah, I like that idea. Yeah. So if just drugs are legal, because I don't quite understand why most of these drugs are, I mean, unless it's like GHB or something, where like the purpose is to create a victim. Most of these, it's a victimless crime. Drunk driving, there's a victim. In certain contexts, when you see the harm to others, there's a victim here. Harm to oneself, there's no victim when you're looking at that. And if unless somebody's doing the drugs to you, you're like, so it's always confusing, because when you look at something like anabolic steroids, like, oh, your liver, well, I don't know, Tylenol's worse. So, but Tylenol, you can just go to S-Mart. Like, what are you talking about? So a lot of the drugs that you can just go down to S-Mart and get these things are worse than this stuff, and this is like you go to prison for. And so there's fascinating commentary, but we'll talk about cannabis from the perspective of its mTOR interactions. And what's sort of weird is the data conflict. So I don't really have too much solid information to give you, but I'm going to give you the state of the evidence, and so that you'll be more confused coming out of these lectures than you are going into them. Now, part of the reason this is so confusing is here's 2016. This is, as far as I know, the latest legality of how this is received. And all 50 states, no matter where you are, all 50 states have some version of legality, accessibility. There's loopholes and there's permissions that are granted here, but the federal government does still regard both CBD and THC in the same class, and it gets a little bit more confusing. I don't know who's writing these things, because whether CBD comes from hemp or its cousin like marijuana, like things get categorized differently, but it still has federal. It's like federally regulated in this weird, unnecessary way, and every state has its own individual policies. Now, down here, so let's just say this is a nerve. Okay, this is a cell. There's cell surface receptors on here, and so that's where bradykinins are going to bind. Prostaglandins are going to bind over here. Track A, tropomyosin receptor kinase A, or tyrosine receptor kinase A, you can call it either one. That's where nerve growth factor would bind. Now I'm going to talk about track B as we get into CBD. I'm going to talk about track B, tropomyosin receptor kinase B, or tyrosine receptor kinase B. Again, you can call it either one. An AMPA receptor, so that's just letting sodium ions depolarize stuff. Over here, you see cannabinoids. This says cannabinoid or opiate receptor. Now, an opioid as compared to an opiate. Opiate is just natural. Opioid is synthetic. So they're binding the same thing. Just like you have endogenous androgens, so the androgenic hormones, testosterone. Dihydroepiandrosterone, dihydrotestosterone, androstenedione, these endogenous anabolic steroids. Your body makes them. Males and females both. Just males make way more. And then there's also exogenous steroids. And so, whatever the I don't know, people are getting in a deodorant container from Mexico and bringing it, whatever, like the illegal stuff, the controlled substances, those are the exogenous steroids. So that's opioid versus opiate. Opiate is natural. Opioid is this synthetic thing, but they bind to the opiate receptors. And it just makes it look like that cannabinoids and opioids are binding to the same thing. That's not true. They're just having a similar effect here. And so you have a cannabinoid 1 receptor, you have a cannabinoid 2 receptor, you have opioid receptors. These are very different. THC is great at binding to the cannabinoid 1 as opposed to cannabinoid 2 receptors, but this is adenylate cyclase, an enzyme that converts ATP into cyclic AMP. It has 3 phosphates and it's down to 1, and it has this little double connection on it. So it goes from ATP to cyclic AMP. And then cyclic AMP is a second messenger molecule. It's going to activate stuff. It activates PKA, protein kinase A. Now we talk about PKB, protein kinase B, as this major anabolic hormone, anabolic enzyme. PKA is a major catabolic enzyme. But if we're in a nerve here, we're letting in some sodium, we're depolarizing the nerve. So if we're going to have prostaglandins causing us some sensation, causing us some unpleasant sensation, they are activating adenylate cyclase. So prostaglandins bind. Remember, so this is our phospholipid bilayer right here. And we have all these cell surface receptors that are going to bind to this stuff, to the cell surface. Now, a steroid hormone can just kind of wiggle itself in and be inside of the cell and have an intracellular receptor. This would be a fatty acid. A prostaglandin is a fatty acid because it comes from arachidonic acid. Remember, arachidonic acid, there's a bunch of cholesterol in here. There's fatty acids in here inside of a phospholipid bilayer. Arachidonic acid is a prominent fatty acid, has 20 carbons in it, and PLA2, phospholipase A2, gets it out of there. Cyclooxygenase converts into prostaglandins. Prostaglandins bind to a cell surface receptor, and depends on where this is happening. But turning on adenylate cyclase, prostaglandins turning on adenylate cyclase. Now, adenylate cyclase activates the PKA signaling cascade. Cannabinoids are doing the opposite, inhibiting adenylyl cyclase and opioids. So here's a bigger look at what's going on. Let's just say this is some like nociceptor. It's a pain nerve. And let's focus in on a few specific ones, stuff that you might recognize here. Histamine, right? So what do you take your Benadryl for? You're blocking histamine. It's an antihistamine. There's your prostaglandins. These are a type of prostaglandin receptor. Oh, CGRP and substance P. A nerve releases those things, right? So the nerve itself, genic inflammation, a nerve-generated inflammation. You're releasing CGRP and substance P from the nerve. Bradykinins, right? There's bradykinins. That's in that delayed onset muscle soreness, right? It's you work out and tomorrow you're sore. Bradykinins are one of them. Prostaglandins really leading the charge here, just like in a hangover. Prostaglandins are a big contributor, but yeah, bradykinins are gonna factor in a little bit. They're going to help facilitate both AMPA and NMDA receptors. So NMDA, you're letting in calcium, AMPA, sodium too, but sodium and calcium, and just sodium with AMPA, and so you're getting ions into the cell. That's a way of depolarizing a cell. Get some ions in there, get some excitatory stimulus, and bradykinins will also facilitate the intracellular release of prostaglandins. So bradykinins are going to contribute a lot to some of this sensation. Oh, ATP, protons, hydrogen ions, right? Capsaicin, the things that make peppers hot. A lot of stuff. But down here, we see cannabinoid 1, cannabinoid 2. Again, number 1, that CB1 receptor is really what THC is preferentially binding to. Some opioids, those receptors, gamma aminobutyric acid, that's an inhibitory neurotransmitter. But lots of stuff is going to bind to a cell surface receptor. The cannabinoids, there's 2 different receptors, right? So THC and CBD, and that stuff is going to bind to that cannabinoid receptors. Now THC, psychoactive, CBD, non-psychoactive. Now stuff affects the hypothalamus and your appetite. Remember all of those F's of the hypothalamus, Van Ness's F's, food and fluid and fight and flight and all those things, all those F's. Appetite is going to be controlled in the hypothalamus, and insulin, leptin, these things are going to shut off appetite in the hypothalamus. But when you see cannabinoids, this is in the hypothalamus, right? Cannabinoids are going to turn on AMPK in the hypothalamus. Now we know what AMPK is: adenosine monophosphate activated protein kinase, AMP activated protein kinase. This is the thing that resveratrol is helping to activate. This is the thing that's inhibiting mTOR. This is the thing that's turning on protein degradation. Now it depends where AMPK is active. Depends where AMPK is turned on. If you're in the hypothalamus, this is the stuff that's regulating appetite. Do you have the munchies? If you turn on AMPK, you want to eat. Turn on AMPK and food seeking and food chewing behaviors. And oh my God, so delicious. Happens in your mouth, right? That's if you have a bunch of AMPK in your hypothalamus. So ghrelin turns on AMPK in your hypothalamus. Leptin turns off AMPK in your hypothalamus. It gets a little bit different when you're out in the periphery. AMPK in the periphery is going to be different. In the hypothalamus, AMPK is a big regulator for appetite. So you do THC, you get the munchies, you're turning on AMPK in the hypothalamus. That's what's happening. And so that stuff is affecting your hypothalamus. Now, out in the periphery, your muscle and everything lives out in the periphery. You will see some differences. Resveratrol turning on AMPK, leptin turning on AMPK in the periphery. But over here, leptin's turning off AMPK in the hypothalamus. It sounds counterintuitive until you think about it. Like, what is AMPK doing? AMPK is trying to be conservative with metabolic expenses. It's trying to be as responsible with its funds as it possibly can. And balancing your budgets is in part spending. Let's shut down mTOR. AMPK, let's shut down mTOR because mTOR is super, 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 super expensive. Super expensive translating proteins. This is just the price tag of that is outrageous. And so AMPK is like, all right, we're going to shut that down. And so let's stop spending. And then up there in the hypothalamus, go ahead and start munching. Like put some food in your mouth. Let's get some calories back in us. We are trying to be conservative out in the periphery, and let's see if we can get some food in here to balance that budget a little better. So you're not just shutting down expenses, you're turning on income. You're putting income in as well. So AMPK in the hypothalamus, let's increase income. AMPK in the periphery, let's shut down expenses. And so something like leptin, right? This is going to be released from fat cells when it's expanding. When fat cells are expanding, you don't need to keep eating. You've eaten enough. Your fat cells are growing. You're literally, they're growing. Your fat cells are getting bigger. Why are you still eating, right? So leptin gets released from expanding fat cells and says, you can be done eating now. Now there's a lot of inputs for appetite. That's just one of them, is shutting down appetite. But in the periphery, if your fat cells are expanding, go ahead and burn a little bit. Go ahead and consume a little bit. Your fat cells are growing. It's okay to consume a little bit of them. And so leptin in the periphery is turning on AMPK. But the only thing I care about here, cannabinoids, hypothalamus, right? Now, cannabinoids in the periphery, it gets a little bit confusing. The research isn't very satisfactory. So let's look at it. Let's look at it, just a couple of studies, and we'll talk more about these studies on Friday. We'll finish up CBD, just a little more information, because it does CBD. There's known information about what CBD does, or at least demonstrated information about its effect in the heart, regulating the heart rate, regulating the blood pressure in a very healthful way. We'll talk about that stuff on Friday, but it's metabolic effects. Here we have this first day. We're going to look at two studies, and just on CBD, we'll do THC on Friday, just CBD. So this is CBD right here. CBD has previously been shown to deactivate protein kinase B, or AKT, right? PKB or AKT. This is from 2017 when people have started using PKB, but AKT is still this lingering relic of its nomenclature. Okay, so we have CBD deactivating PKB and Jurkat cells. This is immortal cell line. So this is like in vitro. It's not in vivo. This isn't like in your body. I mean, this is like, in glass, literally in glass stuff. Looking at cell lines, and we don't have that much in the way of human data of this stuff. But so they were doing CBD and then looking at the phosphorylation states of PKB, you know what that is, and ribosomal protein S6, you know what that is. mTOR phosphorylates P70S6K. P70S6K kinase phosphorylates ribosomal protein S6. Translation happens at a ribosome. Okay, now we're translating proteins. So CBD is reducing the phosphorylation of PKB and ribosomal protein S6. So for the time course, blah, blah, blah. CBD did deactivate mTOR to some extent. And then just looking at these doses, having this dose-dependent response, cannabidiol, again, that's your PKB, or your CBD. And as you get a bigger and bigger and bigger dose, there's your PKB phosphorylation is minimized. Now, this is mitogen activated protein kinase, extracellular signal regulated kinase. There would be an RSK in there too, but this is a separate anabolic signaling cascade that both intersects and converges where mTOR does, where PI3K, PKB, mTOR does. This is another signaling cascade. And we're going to talk about this one as we get into prostaglandin inhibitors for pain management. Ibuprofen, right? Your NSAIDs. We'll get into this one, but they found it like, oh, sure, maybe phosphorylation states increase a tiny bit with a huge dose. But the mTOR signaling, and down here you see the phosphorylation of ribosomal protein S6 if you get a big dose of CBD, is minimized, and there's a time course connected to it. So what they found was a reduction in mTOR signaling in this immortal cell line of T lymphocyte cells, of it's like an immune cell line. And people use it to study leukemia and stuff. If you've The Immortal Life of Henrietta Lacks, if you guys have ever read that, there's a lot of these immortal cell lines, and this is one of them. So they found that ribosomal protein S6 and PKB were suppressed in response to cannabinoids. In response to CBD specifically. We'll talk about THC separately. Now let's go to a new study. Okay, so this one, 2019, and these are good journals. These are legitimate articles, good journals. So cannabidiol induces rapid and sustained antidepressant-like effects through increased BDNF signaling, brain-derived neurotrophic factor. A factor is a protein. Insulin-like growth factor, right? Brain-derived neurotrophic factor, nerve growth factor. 20 minutes ago or something, I mentioned nerve growth factor, and I said nerve growth factor binds to TrkA, tropomyosin receptor kinase A, or receptor tyrosine kinase A. It's the same thing, just like PKB, AKT, right? Different names for the same thing. TrkA, right? That's what nerve growth factor binds to. And so if you have SEPA, those people who just don't feel pain, they don't have any nociceptors, they put their hand in boiling water and they have no idea, like, oh, I just was fetching the spoon out of it. It fell in. Yeah, but it was bubbling. You didn't see that. I don't know. I didn't feel it. Yeah. So that's SEPA, congenital insensitivity to pain and anhidrosis. SEPA, anhidrosis because you don't sense heat, but SEPA, you have this shitty TrkA, and so nerve growth factor doesn't bind to it. BDNF, brain-derived neurotrophic factor, this protein binds to TrkB, and so there's another cell surface receptor for BDNF, that's TrkB. That's what it binds to. And for brain health, this is the thing that you really care about. This is the thing you want. This is the thing that's becoming more and more prominent in research every year for the last decade or so. Every single year, BDNF becomes a bigger topic. And there are supplements out, all of these like nootropic, whatever supplement, and guaranteed to improve your cognition by whatever. Some of the ones, there's some data about coffee berry, like the little berry. I take it. The increases BDNF levels. There's some evidence here and there for compounds that can increase it, but they're looking at mice, little Swiss mice. And so their findings. So the medial prefrontal cortex, that's what this MPFC is. The hippocampus, that's what the HPC is. Rapid effect of, they take CBD, a robust, acute, but also sustained antidepressant effect. And it was rapid, was accompanied by an increase in BDNF levels. So they gave these mice CBD, and they had an increase in BDNF levels in both the hippocampus and the medial prefrontal cortex. And guess what BDNF does? It turns on mTOR. So this is turning on mTOR in this study. So in that cell line, so this is, one of them's a mouse, one of them's in vivo mouse, the other's in vitro with this like T cell line. And in that cell line, CBD was turning off mTOR, PKB and mTOR. In this one, in these mice, they're getting an activation of mTOR through an increase in BDNF. So altogether, our results suggest that rapid antidepressant like blah, blah, blah injection involves BDNF, TrkB, that's what it binds to, that's the receptor, mTOR signaling. Medial prefrontal cortex, and supporting. They just sort of go on and on with the same stuff that systemic CBD administration seems to have a BDNF, TrkB, mTOR signal. And it gives a little bit of room for, is there a chronic difference versus the acute difference? There's room for commentary there. But again, this is an mTOR activation as opposed to the other one that said mTOR inhibition. So now we have articles that are a little bit in opposition to each other. Now we would have more data if people had been researching this for longer. We would have more data if it weren't a taboo topic. There's a lot of room for information, room for growth, and we have not arrived there yet. So at the start of this lecture, I said you probably emerge more confused than you went in. I assume I've succeeded in at least that much. We will finish cannabinoids on Friday.
For NSAIDs, going back to NSAIDs as a hangover prevention, what's the practicality of it in real life when you're talking about stomach bleeds and from NSAID medication?
Yeah. What I would suggest, as long as if you abuse it, if you're an athlete who's doing NSAIDs, daily, there's COX-1, COX-2, and the integrity of your stomach is dependent on you not blocking these things. Doing it once in a while, if somebody's like a couple times a month, they're going to go have the drinking session, once or twice a month, go do your NSAID session. Where it becomes problematic is like if you're a soccer player whose ankles are messy and or you have shin splints or something like that, and so every single practice, it's like 5 days, 6 days a week you're doing NSAIDs. That's a mess. That's a mess acutely. And Liam, yeah, fine, that's totally fine. That's a mess acutely, and that becomes a mess chronically when you start looking at some the stomach bleeds that you see in these gastrointestinal disasters. And we'll talk about this when we get to the NSAIDs. But one of the potential problems is for the acute part of that is you get a little bit leaky acutely, that you seem to. And so how come NSAIDs aren't even a good performance enhancer? If people are sore and they take NSAIDs, they don't perform any better. Tylenol, maybe they perform a little better unless their liver is like, if they have like if they're in the hospital, they're not going to perform very well. But why do people not perform better if they're sore and NSAIDs relieve that soreness? Doesn't do anything for they don't get any better in terms of running time, running speed, whatever the performance is. Like, why did you do that? You're more comfortable performing badly. But yeah, so leaky gut, you get this acute leaky gut. So there's a lot of problems with it. But to answer your question a little more directly, once or twice a month, you're gonna, get ready for a hangover, go ahead and throw in some prophylactic NSAIDs and you'll be okay. If it's like, okay, I drink on Wednesday, Thursday, Friday, and Saturday nights, okay, let's come up with, try some prickly pear sometimes. Let's try to come up with, go ahead and try that B6 solutions. Let's see if that works for you. I would try some alternatives. Other questions for now? Okay, so your homework is to do a ton of THC and go exercise and see how you feel. So we have something to talk about after class on Friday. No, you don't have to do that. It is, there is, I don't know if you've ever done that before, the hyper focus, that like every rep is like the only thing in the world happening. It's what I imagine people are feeling at a slot machine. Like the people who have like those like troll dolls and their little good luck charms and stuff. And they're like pulling slots. I mean, there could be warfare going behind them. And they have no idea because there's just one pull after another. I mean, it's just like the little dopamine surges, and nothing could distract them. I think a THC bicep curl is a little bit like that, where there's nothing else that's happening in the universe. So again, contexts with this stuff. All right, that's all the words I have for today. See you guys in a couple days.